Welcome to the MOOC Interactomics course. In our last lecture, we learnt about the basics of diffraction-based sensors from Professor Cynthia Go. Today, we will continue with our discussion on this approach with her. She will demonstrate various examples for measurement of molecular interactions using diffraction-based biosensors. So, let us start by viewing an example for direct and label-free measurement of anti-digoxin binding to digoxin. We will talk more on the how to measure protein interactions. Welcome, Cynthia.
Thank you. Okay, here I'll just show you an example. Here's an assay. These are results from an experiment in this setup that I showed you earlier, that little prototype. So basically looking at the intensity of light as a function of time as we introduce anti-digoxin to bind to digoxin, which has been immobilized on the glass surface. Okay, so you can see that when the analyte, when the anti-digoxin is 200 micrograms per mil, you get the uppermost curve. As you decrease the analyte concentration, you get the lower and lower curves until down to 200 nanograms per mil. And here on the right-hand side, we blow it up so that you can see 200 nanograms per mil. Still pretty reasonable. I can still believe that's still signal above noise in there.
Noise, yeah.
Yes. So we can go from 200 micrograms per mil to 200 nanograms per mil label free in 20 microliters of solution in this experiment.
Right. I think that's quite good, but I think probably we can tweak it around to enhance the signal.
So this is still, of course,
label-free.
This is label free.
Right.
It also is in that little breadboard setup I showed you earlier. So not optimized in any way. Mm-hmm. In fact, some of these experiments we had done earlier just on a webcam with a manual capture of the intensity.
Right.
So now there's many ways of pushing it higher, but one way of doing so is by putting in a label. I know this is a label-free course, but you can see what a label would do to you.
So I think broadly we are discussing about different type of detection systems. So I think it will fit.
That's right. So you remember that we have a 200 nanograms per mil that was in the previous slide. But now I'm going to introduce a secondary. Here's a secondary antibody, and in this case, labeled with a little piece of gold. Actually, we found out later we don't need that gold label. But anyway, for this graph, that was had a gold label in there, and we can bring it down from 200 nanograms per mil to 2 nanograms per mil. And now this is now this noise is now the noise of the detector. So we're in order to go better than that, we have to do a little bit more of signal averaging.
Sure.
And we can do a different type of labeling, which is this is a precipitation assay. So here's our initial antigen, and then you put in the antibody, digoxin, and then you put in the secondary, which has horseradish peroxidase in it. The horseradish peroxidase then acts on a substrate TMB to form a precipitate. Okay. If you do that, we can actually go down to 50 picograms per mil.
So you achieve much higher sensitivity.
From 2 nanograms, here's 2 nanograms per mil here. Now 2 nanograms per mil is huge, right. And we go down to 50 picograms per mil. I think one can actually even tweak it further, but you're starting now to fight kinetics because if you're very low, of course, you now, this binding is taking a very long period because it takes some time for them to find each other. Yeah. So, but anyway, just in
this could be useful, I think, when we are talking about very weak protein-protein interactions or different type of analytes which are very, very low. That's in abundance. So
yeah. So that becomes now an issue about assay development. What we have here is a tool that allows you to measure that signal. Now you can configure your assay so that you introduce. In this case, we were just introducing them linearly. Right. But in some cases, you can actually pre-mix a cocktail and then let it bind together, and that may sometimes work better.
Sure.
So it's actually not just protein that we can actually look for. These are some data on troponin. This is still a protein antibody assay. The clinically relevant level is around here, above this line, above 2. And you can see this is the signal. This is indicative of the noise of the system. That's why there's wiggles in that signal. But the interesting thing here is from the point of view of clinical diagnostics, that's 10 minutes here. So in less than 5 minutes, you already have a difference between higher levels, clinically relevant levels, and clinically, absence of troponin.
So in a very short time, I think you're able to measure the signal with high intensity.
That's right. So imagine that this is a marker for cardiovascular disease, a marker for stroke, then you can know within a few minutes that it's there or not. Here's an example for how you can actually assay for a couple of antibodies for TB. Here's looking at the 16 kilodalton. So there's, we're just going to show you, in this case, an example of two markers, two antibody antigen binding for TB. Again, it's just looking at two different spots simultaneously happening. And the introduction of antibody to the 16 kilodalton TB antigen. Again, you start with the experiment here, and
so how do you correct for the baseline in the beginning? Because I see it's beginning from 800 or something.
Yeah, so there's a baseline. Oops, sorry about that. There's a baseline here because there is a diffraction pattern. We've put down the 16 kilodalton antigen on the substrate.
Right.
And so when you shine light on it, there's going to be a baseline.
Right.
Okay? And to actually reduce non-specific binding, we put in a BSA block. And so basically here, so that's so that your medium will not have all the proteins sticking to the lines.
Sure.
So you introduce a BSA block.
So first you do the blocking of the surface, and then
That's right. So here, so the signal drops to 0 because of the blocking, because the BSA sticks in between the lines.
Right.
And then at this point, we introduce the 16 kilodalton antibody for TB that will bind to the antigen. And you can see there's a little blip in here. Now, let me expand this area. You can see the expansion here. That blip is actually real because the signal to noise is good enough.
Yeah.
But in case you basically want something that's much more obvious or something that you can see, because at this point, when you have the precipitation, you can actually see the signal already, okay? But yeah, we introduced the HRP goat anti-mouse and the TMB substrate here, and you can see the enhancement in signal. And so this is actually a very big intensity change. You can look at it. This is a 3,000,
That's right.
reading at 3,000, so it's a 3,000% intensity change.
No, that's I think sort of clear yes or no answer, right?
That's right. Yes. So if you're trying to create a diagnostic, just say yes or no. Well, here I can say yes or no. Okay. Yeah. So that's one of the spots. Now, if you look at the second spot, which is filled with a 38 kilodalton TB antigen, you can do the same experiment, and then we'll put them together. Right, there's two spots together.
That's right.
At this point, we introduced the 16 kilodalton TB antibody, and one of the spots got more intense, the other one did not. And then at this point, we introduced the 38 kilodalton, and then you get the other spot got more intense. And then we amplified them both, and both spots.
The blue one is 16 kD, and the 38 kD is the red one. And
That's right.
It is able to measure simultaneously both.
That's right. That doesn't.
And if we see the signal of both, then I think we know that person is actually positive for that.
Well, you have TB is a very hard thing to detect,
right.
but at least now you know that you have two signs it's there, and you can imagine adding, because there's other types of potential markers for TB that we can just add to that, and so you get more and more sure that actually something is happening.
It's more sort of, I think, proof of concept which you are showing, but I think it can be applicable depending on the context.
Yeah.
One can make the assay more robust.
That's
with
right.
the introduction of right type of proteins and more markers.
Yes. Now, the other way to do that also is, you can imagine making the second spot be a blank.
Right.
And that way you can make sure that the signal is, in reference to a blank,
That's
and
right.
that you don't have false positives in there. Okay. We can skip the other ones that other than proteins, but just to show you that you can look at cells, you can look at polymers, because actually the measurement of interaction is, it's general. You can measure the interaction between two types of molecules.
Okay, so I think now you got a prototype, and probably you can discuss that how you took the prototype and actually took it at the commercial scale, because ultimately you need a lot of applications to develop.
Right. Well, I showed you what it looked like in my lab. So, and I can teach you how to make one of those. But unless you want to be an expert in optics, then you don't really want to have to build one every time you do a measurement.
Right.
I could have just keep cranking out data, but that's not the point, right? We want to be able to create an instrument that will be useful to other people. And if we're going to go for medical diagnostics, it's important that the instrument is functional and useful for others. And this is the role of commercialization. So at that point, I actually, around January 2002, or so, basically, I decided that we have to commercialize the device in order to get it to be useful by others. And this is important, that thing that I believe in, that for science to be able to benefit society, it has to be turned into a product that others can actually use. And so this is the path of the instrument. This is how it was in my lab. And up to now, we still have these instruments in my lab. We still work on the benchtop. But what we did is we translated it into a series of steps so that the current device that went out in the market late 2006, early 2007, is an instrument that other people can use. And that little piece of double-sided sticky tape, it's not really going to be good for you to just have to make that yourself. It's now a little piece of plastic that's actually a lot cheaper. It took a lot to get there, but it's now much more efficient and cheaper. Okay, so here's the instrument. It's all computerized. There's pumping system inside. There's software. And it has the little sample cell that has 8 spots in it that you can put down different proteins or different snippets of DNA or whatever biomolecule you're trying to assay.
So now you are providing the multiplexing capability for...
Yes, it has multiplexing here. Instead of on top of each other, it's now in 8 different spots. And effectively in the instrument, the laser beam runs through back and forth into this. And there's a channel there. There's an insertion port, the injection port where you inject the analytes. Right. And it just goes to that little channel, which is about 20 microliters or less.
Yep.
Okay, but it has a pumping system, so you can actually control the flow rate and so on, which becomes important when you're doing kinetic studies. So here's an example of a study on the binding of a protein to a DNA. So in this case, this is the substrate. We put down streptavidin in a pattern, and we basically take that substrate, and here's the initial signal from that substrate.
What is the y-axis here? Are you related?
This is a time versus intensity. This is just intensity in arbitrary units at this point.
Yeah.
Right? So Rec-A is a protein that binds to the DNA, and that's what we wanted here to see is the kinetics of binding and unbinding.
Right.
So we start with a substrate that has only streptavidin, and when you introduce Rec-A to that sample cell, nothing happens.
Right.
Rec-A doesn't bind. So at this point here, we introduce a biotinylated oligo.
Right.
And so that binds to the streptavidin, and you get a little blip and signal.
Right.
Okay? When you introduce Rec-A at this point, you get a big
Much
increase in
greater
signal.
Right.
Okay. And then we want to unbind Rec-A, so we introduce just the buffer to flush out the Rec-A, and the signal goes down. That's because the interaction between Rec-A and DNA is a lot weaker than antibody-antigen interaction. And you can repeat the experiment, etc. So, and you can actually analyze these curves to measure the binding and unbinding kinetics of Rec-A and DNA.
So this will be similar to what we're talking about in SPR, right? Like in terms of on rate and off rate.
That's right.
I think similar.
Yeah, so this is an on rate and off rate of Rec-A onto DNA. Now, similar both in here and in SPR, one has to actually be put together the correct model in order to extract the correct kinetics.
Yeah. So I think the softwares play a role over there, like how you best fit the model and extract the data for.
Yeah, so I think I know that the SPR instruments come with associated software. Now, if you're actually trying to study a specific system, it may or may not be the right model for your system. In this case, you can actually write down the equations and do your own fitting of the data. So that's in order to extract real quantitative kinetics. So in this case, as long as we're reasonably at a certain range of concentration, the intensity is linear with concentration, and therefore you can actually model the kinetics nicely. This is a similar experiment, and this is on the binding of polymerase, RNA polymerase, to the immobilized oligo.
So it shows the kind of different level of applications one can achieve, not only just a strong antibody and protein, but also protein-protein or DNA-protein.
That's right.
Or polymers or cells.
Cells, yeah.
So I think
yeah.
that's
Cells are actually very easy because cells are big, right? So, in fact, the challenge with cells is we have to redesign the lines because the lines, the lines originally we have are about 1.5 microns. Okay. But cells are bigger than that, and so we have to redo the whole thing with a much bigger line.
So different setting has to be.
But it's the same principle, and in fact, it's actually very simple to detect cells.
Sure.
Yeah. So this is just again, this example is antibody immunoassay, so you can play games on that.
And what you have on this one?
So yeah, so this is just antibody quantitation and showing a slightly different surface. This is avidin with a GAM FC in there. It's not different from the other one, so I'll pass by it. Sure. Here maybe is a more interesting example, and I think this is one of the early clinical example using now the instrument. As I said, now the fact that there is an instrument that somebody else can use, that means people can actually configure their assay and play around with how you're going to get it to work best for measuring whatever it is you want to measure.
That's
okay?
Right.
So this is the detection of neuron-specific enolase, which is basically associated with traumatic brain injury. And in this case, Dr.
So is this kind of a marker which indicates the traumatic injury?
That's correct, yes. And I'm not an expert on the subject, but Dr. Berger was using this primarily to look at markers in babies. When babies have shaken baby syndrome, they actually have issues about whether the baby has been shaken.
Okay.
And in this case, being able to use only 20 microliters is very important because you cannot get too much blood from babies.
Right.
And so here's an example where they actually have it's effectively the same as we've been discussing before. You put down one protein that identifies another protein, and sure enough, it works. I mean, as a physical chemist, I feel like, well, if it works for one, it has to work for another. It's a question of how strong the signal is, which is dependent on how big the sample is.
But challenge here will be like in terms of the level of the protein, right? In the different patients.
Yeah, what is a significant level? So in designing biodiagnostic instruments, one has to always ask, if I'm detecting for disease X, what's the relevant concentration to detect?
Right. And whether I'm able to capture the dynamic range.
That's right. Well, or whether it's even important to have dynamic range, okay? So if you're actually looking for a marker that's, present in micromolar quantities, that's not a challenge. You can use anything that you have, right? But also then there's no sense building an instrument that can go down to nanomolar if all you're detecting is micromolar.
Yep.
Now, for many, if your interest is primarily in terms of kinetics, then you don't really run kinetics at low concentration. And so, you don't have to do an instrument that can do nanomolar if you're trying to actually do kinetics, because usually you run that at micromolar. So here's an example of troponin detection. So the interesting thing here is being able to detect a complex system. So in this case, and this is the work of Professor Jenny Van Eyck at Johns Hopkins, again using the instrument already, the commercial instrument. In the surface, you put down the antibody, the anti-troponin. Right. In this case, troponin is a complex that has 3 parts, and you can identify whether all 3 parts are present or in what amount are the 3 parts present by putting now an antibody for each of the parts of troponin and measuring the response. So it's almost a multiplex assay, except for it's all on the same protein.
Same protein.
And again, so at each point here is the introduction of another thing. So the introduction of the troponin, and then the introduction of all the different antibodies to the different parts of it. The way we configure the instrument is actually, in order to introduce a different solution, there's a little gas bubble,
Right.
and so this gas bubble marks where you introduce. It's a time zero. So if you're doing a kinetic measurement, you need to know exactly where time zero is, when things were introduced.
I think in this case, since I think they know the biology of it well, so probably measuring different components was easy, because I think they were able to generate anti-CTNL and
That's right.
anti-TNC and TNT fragments, and then specific antibodies for it. I think it's a pretty neat experiment where we can actually measure the complex protein, but
That's
also
right.
its individual components with
Yes.
their specificity.
Yes, and so you're being able to do that, and then configuring your assay, because you can actually do displacements and take them away.
Right.
And this can be done in how long is this? So this is 5,000 seconds, but it doesn't have to be even 5,000 seconds. If you're not doing kinetics, you can cut this thing shorter,
Right, you don't
right?
need that.
So it doesn't have to be that long. The comparable technique is to actually do a Western blot, and you know how long Western blots take.
It will take whole day,
Yeah.
in fact more sometimes.
And a lot of work and a lot of material. So that was actually a very nice way you can actually do experiments faster.
Sure.
There's various applications that different groups have studied, and this is about detection in PSA. And some interesting work were done on antibodies for isotyping and avidity experiments. So as I said, once the instrument is commercial, I don't even know what these things are anymore.
Obviously. It's just measuring interactions of different biomolecules, and one can use the same principle.
Yeah, well, you can use the same instrument, but now you can configure your assay to get you the numbers you want. Because, for example, if I told you that we work best with large molecules, well, how do you start doing small molecules? Well, you have to be more clever at it. You might have to do a displacement assay.
Yeah.
It's still, it's label free, but you're
Right.
going to have to do a displacement to compare the strengths of interaction.
Right.
In this case, to look at isotyping and avidity, it's about a displacement that they're
It's
doing.
same for all technologies, right?
That's right.
Yeah.
But you see, I personally couldn't have done all these wonderful applications.
Sure.
So again, I go back to the desire to benefit society by sharing your science, and it's not just about publishing it, but actually creating a device that facilitates other
Which can
studies.
give rise to multiple applications depending
That's
on
right.
what user
And
wants.
hopefully get it to be more useful quickly. I mean, if we can have diagnostics that are useful quickly, that's great.
Yeah.
Let me just show you how we actually did multiplex. I kind of glossed over. I said there are 8 spots in there. But to put 8 different proteins on a piece of glass slide is in a pattern is actually non-trivial. And you can do it by physically doing them one by one, but that becomes very, very expensive and prone to errors. So one approach that we've actually done together in collaboration with Axela, the company, is to use what's called the Beckman-Coulter A-squared linkers, which are short snippets of DNA that they actually have created in order to bind to proteins, but are distinct enough so that each snippet is on one location. So, for example, when I bind this dark blue single-stranded oligo to the protein, it will specifically bind to this site.
Right.
So it will hybridize to a specific location. So it makes it a lot easier to, you have to react your protein with this DNA, but that chemistry is known, and then you just inject them all and they go to the right locations. And
So I think you can achieve more interactions simultaneously, and
you can measure 8 different
8,
things at the same time. And that's the whole idea. So the instrument works, and hopefully other people will start developing more. Right now, there's the main people using it are in the diagnostics area, trying to actually develop ways of detecting certain illnesses. But it can be used for research to actually look at the binding kinetics.
yes, I think now since we have developed this device, so now one has to think like where next, right?
Where
So what
to?
is
Yeah. Well, of course, the dream is point of care diagnostics for many, right? Basically, to be able to actually get yourself diagnosed without having to have, lots and lots of vials of blood extracted, sent to the clinic, sent to some laboratory, and to take. So if you can have diagnostics in the doctor's office, you can know quickly. That's a dream. There's a lot of issues in it, and it's not scientific. It's a question really of business and policy and so on, right? So that's the goal for the company.
In terms of detection systems, obviously you are looking at diffraction-based system here, and you're aware like the optical-based system and different type of other platforms are available. What will be your perspective, like where the field is progressing, and what do you think, like is there a specific way one can just select a platform and move forward on it, or depending on our kind of application, one has to see
No,
the
I don't think there is one answer to everything, because different devices have different strengths. Okay, so for example, what is the weakness for something like a diffractive optic? Remember, I told you there has to be repeats.
Right.
So therefore, we cannot detect one single molecule. Okay, so what is the best sensitivity you can achieve? Well, I don't know the answer to that, but it's certainly multiples of molecules.
Right.
So if you're trying to go very, very low numbers, that's not a way to go. So it works best. Probably its main advantage is in a detection of multiplexing, that needs multiplexing, particularly things that needs a wide dynamic range.
Okay.
So for example, if you're looking at cardiac markers, some are present at nanograms, some are present at micrograms. It's very hard to find a technique that will span a very wide dynamic range. But there are other advantages of different techniques, and one has to consider also the ease of use, the cost, the time needed, etc. So sometimes we scientists tend to concentrate on signal and sensitivity, which I still put it down. We want to have better signal, higher sensitivity. We of course want more accurate diagnostics, but then again, just how accurate do you need it? I mean, basically, usually when you're 90% chance of having a disease, probably you'll get treatment for the disease. But from the basic science perspective, we would like to understand kinetics a bit better. As I said, there are models, there are software where you can press a button and out pops an answer. And sometimes you have to ask, well, is that really what's happening? Because once you start to deal with confined media, very small volumes, and you start having other issues, especially if there are other things in solution that you have to take into account.
Do you foresee your diffraction-based sensors in terms of integrating it with more nano elements, some nanomaterial, or with the plasmons, or integrating different components for having the better applications?
Well, that's one way of improving sensitivity. So right now we built this to be as cheap and as simple as possible. I told you there are three elements: a light source, a sample cell, and a detector. Okay. The rest are things that you could remove, but it's there, that's nice for research. If you want to increase the signal, then you can put in a plasmonic surface. Actually, there are two approaches that I'm working with colleagues. One is using plasmonics by having gold either on the surface or as particles or on the lines, and the other one is to actually do elements that are vertical, to actually have confined what's called black surface waves. Again, they will make the device more expensive because it's more complex, and probably because it's more complex, it's less robust. But if you can get 2 orders more sensitivity, then you have a chance of looking at early stage cancer. So there's all these pluses and minuses.
So I think the application is to look for the very, very low abundant molecule at very early stage. So thank you very much, Cynthia. I think as you learned from her lecture, the science part of it, how one has to think about the even simple physics principle and then build devices from it which can be applicable for various type of application. So I'd like to thank again Cynthia for sharing your work with us and giving us a very good insight about different type of detection system, including the diffraction-based sensor, and how one can play with the different type of molecules over there and have either label-free or label-based detection for better sensitivity. Thank you very much.
And thank you.
So in these 2 lectures from Professor Cynthia Go, we learnt about the principle of diffractive optics technology, the features and advantages of this approach, and application of this approach in various types of diagnostics and other biological applications. Thank you.